we have been talking about multiple things about, the disease, developing resistance, lot of residue issues, the chemical residues leading to rejection of lot of export material, and lot of climate change related leading to lot of biotic and abiotic stresses. And can we follow integrated pest management or integrated crop management? So to have these kind of a, to address all these major issues, we need to have sufficient tools with us so that, we can integrate and also, the consumers, the farmers, the government agencies, policymakers, everybody wants to reduce the chemicals by 30%, by 50%, but do we have enough tools to really replace chemicals? So to address these, I have picked up a subject, which can really support the integrated crop management and address both abiotic and biotic stresses and still give very good yield under extreme conditions or even, in the presence of many pests and diseases. I'm talking about, the plant immunity inducers. This is somewhere in between, plant breeders breeding for resistance and crop protection experts providing chemical solutions to manage biotic and abiotic stresses. So this is an interesting area of research which has been studied. I think, plant immunity has been studied for quite some time now, 30, 40 years, but the tools are also being used in some cases, but with the advancement of, the science, molecular biology, biotechnology, synthetic biology, things are really improving very fast so that some of the tools, which are available now can be effectively deployed, to reduce both abiotic and biotic stresses and promote the growth and get enough yield. You all may be aware the biotic stress and abiotic stress contribution to the yield loss, if biotic is causing like, 50% loss, due to biotic stresses, the abiotic stresses are causing more than 70%. Means, the contribution of abiotic stresses, which we may or may not be aware, abiotic stresses are causing more yield, that's more yield losses. That's why, today our average national yields are so low. So in this direction, let us quickly understand, the entire, the plant immune system. We know that, plant has to defend itself through its innate, innate immunity to defend against, incoming pathogens. Pathogens, when I say bacteria, fungal diseases, viral diseases, a lot of fungicides are there to manage fungal diseases. Now, bacteria and virus, the big crop protection industries, they don't spend a penny to come out with the solutions. So in the crop protection industry, when we talk about virus management or bacteria management, these are orphan diseases. The big industry, the big 10 crop protection companies, they don't spend any money on inventing a new bactericide or new virus management tool, which means we need to find alternative tools to manage these diseases. Means, if there is a bacterial disease today in any of the crops, including chili, do we have any good solution after banning antibiotics? The answer is no. So in such situations also, the immunity, the innate immunity can take care of these diseases. So how it works? You know that, unlike, the plant, unlike the mammals, wherein, you have adaptive immunity and moving, B cells or T cells to fight the incoming, foreign pathogen, the plant immune systems, are little, not so well, or they don't have these adaptive immunity system in the plants. In the absence of that, but, there are a couple of important concepts like, there are patterns, molecular patterns of the pathogen or on the microbes or on the nematode, which is coming and, infecting the plants, and correspondingly, there are, receptors on the plant which can identify these these markers or these these that okay, we have moved one slide back, please. Yeah. So there are MAMs and PAMs or dams on the pathogen, and correspondingly there are the receptors on the plant. The recognition happens. Once the recognition happens, there is a signaling, there is a lot of signaling happening in the plants, which leads to a lot of pathway activation or gene upregulation, through which, several hormones like salicylic acid, jasmonic acid, ethylene, all these, pathways are activated, and these hormones are accumulated, then which will lead to, oxidative burst, that is ROS production, or sometimes, superoxide, hydrogen peroxide, RNAi. There are many, many responses for this, the pattern triggered or effector triggered the immunity. So means, there is a set of process by which plant can defend itself for incoming pathogens. So the next one, yeah. So in this, two concepts are very popular. One is SAR, another is ISR. SAR is, systemic acquired resistance, which means, there is an incoming pathogen, or those patterns from the molecules or effectors, or, sometimes you would have heard about harpins in plant pathogenic bacteria. These are the real effectors. Because of the production and introduction of these into the plant responds by identifying them and integrating and increasing or upregulating some of the pathways. Means, the ISR happens when there is an infection from the pathogen, whereas ISR is mere presence of some of the beneficial microorganisms in the soil. There are many, many microorganisms, like including the trichoderma, there are some pseudomonas, some bacillus, all these mere presence there will induce the resistance. This is called induced systemic resistance. Means, in both the ways, the resistance is acquired, sometimes very quick. With SCR happens within 4 to 5 hours, whereas ISR is little long-term and broader. ISR happens over a period of time. Both are systemic in nature. So what is important is, in SAR, the salicylic acid pathway is activated. Further reaction to that is like, PR proteins, or, there are many things, callus production, lignification, and RNAi. Many responses will come from the plant. Whereas in ISR, the jasmonic acid pathway and ethylene, hormones are produced, and further, the next response from plants will be similar, in both. So SAR and ISR today are being studied very effectively. In some cases, they are combined so that, the plant resistance is durable, as well as, acceptable control is achieved. So means, multiple plant response can be there for a pathogen which is, getting into the plant, and at times, if the timing is right. When I say timing is right, you have treated the seed with, SAR or ISR molecule or plant immunity inducer, or you've gone and applied these molecules in the nursery itself, then the response of the plant in the entire cropping period is wonderful in two ways. One is, okay, I am using these plant immunity inducers upfront through the seed treatment or in the nursery treatment. What happens is, it delays the occurrence of disease. Very important. In all virus diseases or plant diseases, if there is an early infection by leaf curl virus, the losses are to the tune of 100%. If the same virus comes after 65 to 70 days, farmer can still get some yield. So early application of these things can delay the occurrence of the disease, and also when there is a low pressure, especially in the polycyclic diseases, you go and apply, and your initial inoculum is so low, even a minor insecticide, a fungicide, or a bactericide, whatever the other plant protection measures will work very effectively because the inoculum load is so low. Means, in a way, these are real good tools where you can really reduce the crop protection chemicals by up to 30 to 50%. This has been the result of many trials across the world, especially in North and South America, where the usage of these molecules is very high. And next slide, please. Okay, where are these molecules? Are they commercially available? Yes, there are many commercially available plant immunity inducers. In Japan, the chemical plant immunity inducers are very popular in rice. You know that in Japan, rice is God, rice is lifeline. For evening, for the day, I mean, in the morning they eat rice, in the evening they drink sake. So, means, without rice, that country doesn't exist. So, in rice, there is a blast, rice blast. Today, rice blast in Japan and Korea are managed 100%. I'm talking about 100% managed by ISR chemicals, not biological solutions. There are 4 chemicals which I have listed: probenazole, isotianyl, tiadenyl. These are the molecules, these are chemical entities which they don't kill any pathogen. They don't kill fungi. They don't kill bacteria. But they enhance the SAR, systemic acquired resistance, which means there are chemicals which are working very effectively. Please note, in Japan, all these chemicals are loaded in the nursery box itself. They put all these chemicals, whether it is a chemical insecticide or fungicide or some of the nutrients, they put everything in the nursery box because they are weekend farmers. They don't go and see the crop every day. They put everything, they plant, once in a while they go to the plant and see, and their yields are superior to us. Means, you can still manage if there is a knowledge, if there is enough, information with the farmers. Coming back to biological solutions in the same space, I think, yes, biotechnological tools are advancing and the science is advancing. Recombinant proteins are coming. The peptides are emerging as new antimicrobial solutions. So there are multiple products. Harpin is one in 1992 in Cornell University. They came out and presented this. The whole world was excited about it. Yeah. The harpins are the proteins produced by plant pathogenic bacteria. So when you isolate these or synthesize these and spray it to the crop or treat the seed, the plant, that PAMPs or DAMPs are there, the markers, the patterns are present in this, polypeptide, somewhere between 20, even up to 100 amino acid chain. They recognize this and consider it as a danger, and the plant produces, or triggers the effectors, the triggers, and enough, the pathways are activated. And okay, there are 2 things in this. One is controlling and boosting the immunity by priming through plant immunity inducers. Sometimes if you continuously prime, the plant energy will go exclusively to manage the diseases, right? Sometimes what happens is, all your energy is gone fighting the pathogen. That should not happen. There should be balance. ROS continuously, if it is produced, plant will die. ROS has to be stopped. I think these solutions which have come in the modern innovation, they stop the ROS or the calcium influx at some point in time, and also they enhance the production of the plant growth hormones, especially auxins, and also the photosynthesis rate improves, so that you have been fighting the pathogen for some time. At the same time, you are not compromising on the yield. Whereas the Bion, the BTH chemical, which is an analog of salicylic acid, which Syngenta launched 20 years back, the problem was it was giving excellent protection against the diseases, but there was a huge compromise. At the cost of yield, there was a compromise. So that will not happen with modern especially harpines and some of the new, sulfated, oligosaccharides, chitins, and peptides. These are the molecules where, the growth and the protection is very well balanced, and at the end of the day, you have protection as well as the good yield. And there are multiple products coming out, especially diseases where it is very difficult to manage. What are those difficult diseases to manage? Bacterial diseases. Citrus greening. So I was talking to Dr. Krishna Kumar about, the management of citrus greening in California. Today, California, they don't talk much about citrus greening because these tools are available and they're using it from the beginning. Trunk injection, spray, they do a very good job, and almost 3-4 molecules which I have listed here, they are in the schedule. Means, all the citrus plants in California are being applied to manage the citrus greening. Similarly, I have seen one of the molecule here, Phytolex, which is ascoroside. It is derived from, soil-dwelling nematode. It's a pheromone. There was a trial conducted in, West Godavari. I can assure you the results were so fantastic, 80% control, whereas Coside was giving 65 to 70%. Just imagine if you combine Coside plus this kind of a solutions and time them, and you get the best control. In the absence of antibiotics, this can be a great boon or even, a good tool in the integrated disease management. Viruses. With this harpin, the harpin is like, though it is from a Cornell University, and later it was commercialized by a private company called Plant Healthcare, and this Plant Healthcare has been acquired by an Indian company now. These solutions will be available for majority of the farmers in couple of years. It's just arrived, it is registered on sugarcane. It's matter of time, these products will be registered broadly for the virus management or bacterial diseases management. So when such solutions they come, like, if you can integrate these solutions and grow the crops so that, you reduce the usage of chemicals, means you are skipping 1 or 2 applications, which means the resistance development is delayed. Residues, if you reduce the total chemicals, the total residue on the crop goes down. The pressure is less, it goes down. Sustainable solutions. So these solutions are looking very beneficial, and I'm very sure you will see them. Next slide, please. Okay. This is another interesting fact happening. All the French seed companies, that France exports a lot of vegetable seeds across the world, maybe number 1. All the French seed companies have come together and formed a company called Succeed. Succeed means, the only job of Succeed is to find alternates to chemicals and still achieve, a priming molecule and a defense molecule and protection molecule which can go on the seed and still get very good, protection of the seed as well as the seedling. So in this, they are doing great job in terms of identifying the RNA, short RNAs, and also the peptides and microbial engineering. All these they are doing so that, this can be used. And the interesting finding from their trials is they are treating the parents to get beneficial results on the F1s, which means, you treat the seeds or you spray certain things on the flowering, some of the bacteria on the flowering males and females, all that, epigenetical changes which you see, on the F1s. And they have even further said, one step ahead, and they said, the best way to deliver plant immunity inducers is treat the parents, the progeny will perform better. Means, these kind of innovations are coming up so nicely. I want especially the seed companies to look at this as an opportunity, to make certain changes, especially, the epigenetic inheritance. the transgenerational change if you want to bring, I think there are solutions which are coming up nicely. Only thing is, it's a matter of time, your parent companies, if they are there in France, this data is available. Last, final slide. Okay. Thank you. I think, the message is clear from this. These immunity inducers have a great role to play in the near future and in the future as well, in the protection segment. These are sustainable solutions. These are eco-friendly solutions. And some of the advances which are coming in this area are going to boost, further efficacy and also bring cheer on the farmers. And, I completely agree with Bhagirath that we need to integrate all these tools and reduce the chemical usage, reduce the resistance development, reduce the rejections because of the residue, and these are real climate smart solutions. Thank you. If you have any questions, please let me know. Yeah.
Thank you, Venkatesh, for a very basic and something which is going to make changes in the years to come. A couple of quick questions. Dr. Krishna Reddy, please.
Good presentation, Venkatesh, but your application SAR and ISR is not a permanent. This work is happening since 50 years back. So any SAR, ISR is not permanent, it is a temporary relief. How can we improve the immunity for long lasting using these chemicals? Some of the chemicals you have mentioned also, used even in India, they are all temperature dependent, climate dependent. Japan, you quoted example, because of the climate is totally different from that of ours. That is how it is working well. So how you will justify that they cannot be independently act together? It has to be support system for IPM. That way it will help, but not independently will be solution for our plant health management.
That's the correct one.
Thank you, sir. I think,
Another point, you mentioned the case of citrus greening and the USA. Last year I was in USA with the citrus for 3 days. My friend Manjunath and me, we were traveling. I wanted to see that how the farmers are doing in nursery and all. If you see the Florida, they're using left and right antibiotics, recommended and 3 injections, and so expensive they're doing, but their success is 50%. But they are now come to another solution. This is not helping. Now they are using microbes for managing. Whereas in California side, they don't want to use this because they are failing in Florida, but they are insisted and funded a project for resistance line development. Manjunath's wife, she has bred. Now there are 32 lines in different species of citrus, and within 1 or 2 years they will be ready for resistance. Absolutely greening-free resistance. That's all I want.
okay.
Yeah, there is no second opinion that they are a component of IPM. I think Venkatesh also would agree. They are ephemeral in nature and in combination with IPM,
Correct.
in combination with identifying our own kind of innovative solutions and phytochemicals that are more adapted to high temperature, I think they'll go. So let us not throw the baby with the bucket. Let us adapt to our circumstances. Identify new. Venkatesh?
Let me answer that, sir. 100% right that, this cannot be standalone solutions. I think, we will be overly confident if you say chemical is Ramban or, this is Panacea. No solution is permanent here. What is the intention of this? Integrate these solutions so that you reduce the chemicals. Tomorrow, like Sri Lankan government, ban all the chemicals, what happened to that country? Europe wanted to take a decision to reduce 30% chemical usage by 2030. They did not. They couldn't take the decision because farmers are still important, food is still important. My sincere suggestion is, or submission is, start looking at these solutions as part of the integrated crop management. There is biotic part of it, there is abiotic part of it. If there is one solution which can really balance it, this is the one. Coming back to the temperature dependent performance, that is true with chemicals, not with biological solutions. Harpins are highly stable. At 90 degree, they are highly stable. Harpin proteins, some of the harpins which are coming to the market, they are recombinant proteins. They are not from one harpin from, Erwinia. There are, polypeptides coming together, forming a heat stable harpin recombinant protein. These are, the technologies are advancing really fast. Whether I spray on sugarcane in Karnataka or in Maharashtra or in Meerut, the results are coming same. Technologies are
I'll also
changing
take
fast.
other questions because I want you to answer maximum questions. Keep the answer short.
Yeah.
So, any other questions?
Sir,
Dr. Harish.
this is not a question, sir. In market, we have thousands of such products. Farmers are really confused. One thing is whether they should use them or not. I personally witnessed some of the products are really performing well, but are they science-based is one question, and the message to the farmer should be clear. And all of them, as per my understanding, now they are under FCO, I suppose. Most of them.
You are talking about general category of biostimulants.
Yeah, that's what, sir. Biostimulants, okay. So, the message should be very clear from this forum to the farmer, to the policy maker and to the industry. It should be very precise and clear, sir.
Actually, in a lighter vein, when Harish gets up, I get confused.
No,
I'm just joking. It's a lighter vein, I said. It's a very lighter vein.
Sir. I am talking on behalf of the
me
farmer
too.
only, sir.
Me
Yeah.
too. What I meant is, your points are so at the grassroots level for the benefit of the farmers. They actually tickle a nerve as to what extent we are becoming relevant to the farmer's problem. That's what I meant in a very positive way. Dr. Bhagirath ji.
Sir, very nice presentation. These harpins,
आप थोड़ा
it will fall under what category? Will it go to?
I think, there are category of the registration is what he is asking. In US, it is registered as biopesticide. With all exemptions, because it is a safe molecule, it's a natural molecule. Unfortunately, in India, biopesticide registration takes more money and takes little longer time to register, and people are going with biostimulant and struggling to claim and promote the products properly. Similarly, even UPL has registered their product in the category of biostimulant, trying to sell this product as for downy mildew and powdery mildew management, and they are not able to do it because you can't print publish. So my suggestion to the industry, crop protection industry, is register them in the right category, so that you can claim. Okay, there are difficulties like crop protection products, whether it is a biopesticide or a pesticide, you need to have 70% minimum efficacy. Otherwise, they will not accept it as a solution. I think we have convinced the commissioner that he will look into the case by case, and even if it is not 70% efficacy, like 3 applications, 70%, he will consider it as an integrated management and still give the registration in biopesticide segment. I think all the biological control segments, if government asks 70% control, we may fail, because you can't expect these solutions to provide 70%. There are some difficulties.
Okay, sir.
Yeah.
Just it is not a question. It is some of the suggestions to the farming community. Your presentation is very excellent. I am very thankful, because nowadays we have to manage the disease not with chemicals. We have to manage the disease. We have to make plant very strong. If you know the disease triangle, host is one of the important factor. Pathogen is one of the important factor. Third one is environment. Environment is not in our hand. In our hand, a plant is there and our pathogen is there. We can control the pathogen and also we can make plant very strong. Under such conditions, now the new under biological pesticides, which have recently presented at Raichur University, it is known as a biostimulant. It is a new era of plant plant protections which will induces the plant to make very strong and sturdy against a pathogen. It will not kill the pathogen, it will not 100% control as to that of chemicals. You cannot compare with chemicals. So in market there are so many products are available. For example, when I was in the professor in head at pathology Dharwad, there was one product at that time released by the Syngenta, Isabian. Isabian is nothing but it is a polypeptide, because we know the role of amino acids. There are 14 PR proteins, pathogens related proteins are there. It is the first compounds which is released as a polypeptide. It is available in the market as Isabian. You can wonder, you see the products, lignin is available in the market. I actually, how it is useful? That will all induce the resistance. So similarly, there is a lot of work has been carried out on physiological plant pathology at TNAU. Just spraying with boron, it is already proved that has managed the papaya ring spot virus. How? Because the virus particles enter from one cell to another cell through plasmodesmata. This boron will interfere in the transmission of the virus. This has already proved. That wise, we are all recommending the inclusion of boron along with the pesticides and also the micronutrient. So these are some, there is a lot of confusions and lot of recommendations. So I request the team, there is a lot of research has to be conducted by the concerned scientists regarding the role of biostimulants in managing the pests and diseases. This is one of the important suggestions. And also in the market, chitosan is available, and blue-green algae, laminarin, that is also available in the market. Then your lignin and also Actigard, that is Biocon, they are all available in the market. But our farmer, they don't know. ಅವ್ರು ಏನು ಗೊತ್ತಿಲ್ಲ, ಯಾವ್ದು ಸ್ಪ್ರೇ ಮಾಡಬೇಕು, ಹೆಂಗೆ ಸ್ಪ್ರೇ ಮಾಡಬೇಕು. ಏನಾಗಿತ್ತು ಅಂದರೆ ಫಾರ್ಮರ್ಸ್ ನೋ, ಬಿಕಾಸ್ ಇಟ್ ಈಸ್ ಕನ್ಸರ್ನ್ ವಿತ್ farmers. Because what the farmers, they are in the mind that if you spray that particular insecticides or fungicides, especially insecticides, just by returning to that site, the insects should have died immediately. They will claim that it is a very good pesticides or very good insecticides. So such of the things should not happen in the future. We have to wait for some times. There are different mode of actions. So in this line, there is a lot of research has to be conducted on biostimulants, especially for plant production wing. Thank you very much.
Thank you, sir. So it was a very nice session, except to say that it is a serial. I think today the discussion is over. Tomorrow the discussion, one more session, the serial will continue with many of these aspects. I am happy for the presenters who made the session very interesting, and more importantly, Venkatesh. So I am happy that there was an excellent participation from the audience. I think we are more or less kept with the time, and I thank everybody for participating very actively in this session. Thank you one and all.